The overall goal of this procedure is to provide efficient methods of assessing microbial infection of cardiac cells using in vivo and in vitro models of heart colonization and cardiac cell invasion. This is accomplished by first growing bacterial cultures and collecting, washing, and resuspending the bacterial cells in phosphate-buffered saline. Next, cardiac cell monolayers grown on glass cover slips are infected with the bacterial suspension, or the bacterial suspension is inoculated into mice using tail vein injections. Then, the cardiac cells are incubated in the presence of gentamicin to kill extracellular bacteria, followed by cell lysis to recover bacteria, or infected animals are monitored for well-being 2 to 3 days post-infection, then sacrificed for isolation of target organs. Finally, cell and organ suspensions are diluted and plated using a spot plating method to determine the numbers of viable bacteria associated with cells or target organs. Ultimately, bacterial colonies are counted on agar medium to determine the efficiency of cardiac cell invasion for cells grown in tissue culture, or the efficiency and extent of cardiac infection in animals.
Though this method can be used to investigate the potential for different clinical and environmental isolates of Listeria monocytogenes to cause invasive cardiac disease, it can also be applied to other microbial pathogens capable of targeting cardiac tissue, or the protocol can be modified to investigate other organs or tissues of interest.
The day before performing the invasion assay,